Howdy, Sam Fluid Science, looking at cell theory and a fluid mosaic model of the cell membrane. Thanks to Sam Moyle for producing this PowerPoint. Science understandings we're going to look at. Cell theory unifies all living things. Cell membrane separates the cytoplasm from surroundings and controls the change of materials, including nutrients and waste. And you need to describe and represent the fluid mosaic model of the cell membrane. So we're going to talk about cell theory. Cells first observed by Robert Hooke, or at least described by Robert Hooke, when he was looking at cork cells in the 1600s. With cell theory, we talk about things like cells being the structural and functional unit of life. They are the building blocks that make up all living things. They are the physical structures that make up all living things, our cells. They're also functional in terms of doing the things that living things do. So whatever we talk about living things and what they do, they can refer to cells and also large organisms like us made of many, many cells. So let's have a look at cell theory. So cell theory is a unifying concept. The reason why it's unifying is because all living things can be described by cell theory. So every cell comes from previous cells and contains hereditary information. One of the aspects of cell theory is that all cells come from previous cells, and that information is transmitted from one generation to the next. So there's hereditary information being passed from one generation to the next. This is one of the ideas in cell theory. Here are some other things that we can talk about in terms of cell theory. All living things have an organized structure, and there is complex chemistry going on inside living things. They take in energy in various forms from their surroundings, and they use it to perform living functions. They have an internal composition that's chemically different from the external. So we have a cell membrane that separates the inside from the outside, so the inside is different from the outside, or the intracellular environment is different from the extracellular environment. Living things have to respond to stimuli, they reproduce themselves, they grow and develop, and the smallest thing that can do all of these is a cell. So a cell has an organized structure, it takes in energy and uses it. It has a different internal composition compared to the outside of the cell. Cells respond to stimuli, they reproduce, they grow and develop. Viruses don't fit a lot of these aspects because they can't reproduce themselves. They need to hijack other cells to do their jobs, to make them. They don't use energy from their surroundings. Not everything that we might think of as being an organism fits in with this because viruses don't. So that's a little bit about cell theory. Now we're going to talk about the cell membrane. So the cell membrane is a very important structure found in all cells. All cells have a cell membrane because they need something to separate the inside from the outside, so the intracellular environment from the extracellular environment. It looks like this. So we have these phospholipids, which are these, there's a head and two tails, lots of those. There's a bilayer of those, so they just arrange themselves in this manner. So we have a hydrophilic head and hydrophobic tail. The hydrophobic tails attract themselves, the hydrophilic heads point towards water, and there's water inside and outside the cells. Then we have some proteins dotted in between those, so we call it a fluid mosaic model. So when we talk about cell theory, we talked about how the inside of a cell is chemically different to the outside. And the cell membrane is important because it ensures that this happens, so that we have a different concentration of certain substances inside the cell compared to outside. All the things we mentioned in terms of things like nucleotides, we've talked about energy. We're going to be talking about energy in a lot more detail. We've talked about ions as well. We've mentioned pH in terms of making sure the enzymes work properly. So we need to moderate the conditions inside the cell to make sure it's amenable to enzymes working, and also for the cell to just survive and keep on doing what it does. So we can compare the inside of the cell compared to the outside. So here we have ions, for example. So ions are charged particles. So we can see there is a lower concentration of sodium inside a cell compared to outside, but there's a higher concentration of potassium inside compared to outside. So the way that these get inside the cell will be different because we need to follow concentration gradients. So we're going to learn about that coming up. So cell membranes are pretty thin, so it's only 8 nanometers thick. The model I mentioned before is called the fluid mosaic model. So there's a phospholipid bilayer, so we've got our phospholipids, they're arranged into a two-tiered layer, so one layer down the bottom, one layer at the top. And there's protein molecules that are embedded throughout, so proteins, and there's also other bits and pieces embedded in there too, but it's mainly proteins with other stuff attached to them. It's a fluid because the phospholipids are constantly moving. It's not solid. Those phospholipids are moving, they're bouncing around. It's a fluid, it's liquid, but it's also a mosaic because there's those proteins embedded between it. So that's why we call it the fluid mosaic model. So here's a zoom in of what we've been talking about. So here's our phospholipids. We've got our hydrophobic tails that attract each other because they're nonpolar, and we've got our hydrophilic heads which are attracted to water, which is outside. So hydro means water, phili means liking, so they like water. So they form this double layer, that's why it's a bilayer. Then we have these proteins. So some of these proteins, for example over here, we have a transport protein, Here we have a glycoprotein with a sugar attached to it, so this could be used for signaling, and those glyco bits, they're carbohydrates. And we kind of mentioned these a little bit before as well. So it's a fluid and it's a mosaic. So the job of the cell membrane, it separates the inside from the outside of the cell. It also regulates the passage of substances into and out of the cell so that we can maintain those concentration differentials. I mentioned just before cellular recognition, so cells being able to communicate with each other through things like ligands. So for an example, a hormone is an example of a ligand. And again, we've mentioned this before too. It also allows for the cytoskeleton to attach to something, and we'll talk about what the cytoskeleton is, that's coming up soon. So it's the internal structure that holds everything inside a eukaryotic cell. I mentioned those sugars before, so we've got those sugars that can act as receptors, so cells can recognize each other because of those polysaccharides attached to specific proteins in the cell membrane. So check. Cell theory unifies all living things. Cell membrane separates inside cytoplasm from the outside and controls exchange of materials, and describe the fluid mosaic. So now for Living Science, we looked at cell theory and we also looked at the cell membrane. That's it for Living Science today. See ya.